This is part 2 of the group problems for lecture exam 1. Part 1 contains questions 1, 2, and 3. So part 2 will have the questions after that. So we'll start with 4. Number 4 asks about osmosis, osmotic pressure, osmolarity, osmicity, and tonicity. And then it asks to complete a table that looks at the different ways to figure out the tonicity of blood versus other things. So we'll kind of go through the definitions here. Osmosis is the net movement of water across a semipermeable membrane in response to a concentration difference. Osmotic pressure is the pressure due to the number of particles dissolved in a solution. These are regardless of size or charge. So a glucose molecule has the same osmotic pressure as a sodium molecule. Okay, so size or charge does not matter. Osmolarity refers to the total number of particles in a solution, irrespective of size or charge. And osmolarity really is the measure of osmotic pressure. Osmicity is a term we don't use very often. It's the initial difference in osmotic pressure determined by the total particles in a solution. So it's what happens first. It's before penetrating particles move and things like that. So it just looks at the initial conditions. By convention, it's always compared to the cell. So you can have an isoosmotic solution where there's no net difference in osmolarity, a hypoosmotic solution where the initial condition has a lower concentration of particles, hypo, outside of the cell. Hyper would be the initial condition would have a higher concentration of particles outside the cell. Tonicity describes what happens to cell volume when placed into a solution. It really describes what happens to the cell, but it's in reference to the solution. So isotonic means there's no change in cell size because there's no difference in the gradient, the osmotic pressure inside and outside the cell. Hypotonic means the cell gains water, right? It swells in that solution. So the solution has less particles, hypo. And hypertonic, the cell loses water. It shrinks when it's placed in the solution because the osmotic pressure is higher outside the cell. It has more particles, which pulls water out. So when you do these problems, there are a number of steps that you need to do. I think thinking about it in 5 steps really helps in general. Step number 1 is you want to make sure you're in the same units. Units matter. So when we look at units mattering, we should have everything in terms of the units of milliosmoles. So that's the MOSM, that's milliosmoles. And that's pretty easy. Just take the millimoles and multiply it by the number of dissociable particles. You are responsible for knowing the dissociable particles and the behavior of the following 5 solutions: urea, glucose, sodium chloride, potassium chloride, calcium chloride. And so here are the dissociable particles. Remember, the osmotic pressure is due to the number of particles. So if I put sodium chloride into a solution, it actually breaks up into 2 particles. So then we have to take that into account. For every 1 molecule of salt I put in, basically, it becomes 2 particles in the solution. So you just multiply the number of dissociable particles. Second thing you do is you determine the osmicity. And the osmicity, again, is the initial conditions of the cell. It's so easy that some people forget about it. All you do is you look at the numbers. Now that you have the inside of the cell and its osmolarity, the outside of the cell, its osmolarity, and you compare the solution to the cell. If it's the same, it's isoosmotic. If the solution is less, it's hypoosmotic. If the solution is more, it's hyperosmotic. Pretty simple. To determine tonicity, the next thing you have to do is you have to decide is the molecule permeable in the cell membrane. Of the 5 solutions that we use, the only one that's permeable is urea. So here we see urea. It is permeable in the cell membrane, which means if we put a cell in a solution of urea that has no urea inside of it, urea will diffuse into the cell across the membrane until it reaches equilibrium. Okay? So the other ones are impermeable, means that they're stuck. Okay? They can't move inside the cell at all. In fact, the red blood cell, the things inside the red blood cell, is stuck inside. So the only thing we have to worry about moving is urea if we have it. Okay? So once we move urea, here's probably a better summary of what I just said, then we just move half inside because it'll reach equilibrium and leave half outside. Once you do that, you have to adjust the numbers though. Okay? Then the next easiest way to do the next step is you have to determine the direction of water movement. And what I think is the easiest way to do is just draw an arrow from the smallest to the largest number. So just point to the largest number with that arrow, and that'll indicate the movement of water. Now, if the inside of the cell and the outside of the cell have the same osmolarity, then you have a double-headed arrow, and there's no net water movement. So once you know the water movement, now you can figure out the tonicity. If water moves into the cell, it's called a hypotonic solution. If the water is a double-headed arrow for its movement, then it's isotonic. There's no change in cell volume. And if the arrow is leaving the cell and going into the solution, water is going to be pulled out of the cell and it's going to shrink, and that's a hypertonic solution. So we'll kind of apply those to these different situations. Okay? And there's a whole bunch of different ways to do this. One way is to just look at the various things. And I'm just going to do a couple of them, and that'll give you a chance to practice with the other ones. So don't look at this too closely until you actually play around and do it. I think we have, what, 9? We have 9 in this table. So let's do the first one. It's pretty easy. So the first one gives me a solution of 280 milliosmoles of glucose. Okay, so that's my starting solution. And I drop a red blood cell in it. Now, don't always assume it's a human red blood cell, right? But if it is, then that means human red blood cells are also typically around 280 milliosmoles. So that's going to be something I want you to know. Human red blood cell has 280 milliosmoles in it. Now, it's easiest, I think, to think about it as 280 particles. It's not. It's actually 0.28 of Avogadro's number of particles. So 0.28 times 6.022 times 10 to the 23rd. Right? So it's this big, huge number. But if you think about it, 280, it's relatively the same as the other numbers. So you just kind of narrow it down to a nice, easy number. That's not too hard to deal with. So it's 280 milliosmoles inside the red blood cell. So you look at the initial osmotic pressure. Okay, so you're looking at the number that is in the solution, and that's 280. The number in the cell, and that's 280. Okay, so we're in the same units, right? Milliosmoles. So number 2 is determine the osmicity. So I compare the solution, 280, to the cell, 280. It's the same. It's isoosmotic. Right? So here's the isoosmotic. Number 3 was move urea if we have it. We're just glucose, right? So look right here. This is all glucose, so I don't have urea, so I don't have to worry about it. So I'm done with step 3. Now I draw an arrow to indicate the biggest, most water movement. And for that arrow, you're going to draw a double-headed arrow, right? Because it's 280 inside, 280 outside. You draw the arrow to the biggest number. It's the same. So there's no net movement of water. Since there's no net movement of water, there's no change in cell volume. And so that means it's an isotonic solution. All right. So let's look at number 2 and kind of do it a little differently. All right, I don't know how well this is going to work, but we're going to try it. So I just want you to note that for number 2, our number is 360 millimoles of urea. That's my starting number. That's my substance. All right, so now I'm going to move to a different part of this that has more of a blank page and kind of play around with that. Okay, so we said that our solution is 360 millimoles of urea. Okay. Now the first thing we do, number 1, is you check the units, and the units given were millimoles. We need to be milliosmoles. So to get to milliosmoles, we multiply by the number of dissociable particles. In the case of urea, it's 1. I put 1 molecule urea in the solution. It stays as 1 molecule. So this one's easy. I just times it by 1, and that gives me 360 milliosmoles. So 360. It's hard because I'm writing with the mouse, but it gives me 360 milliosmoles. So my solution is 360 milliosmoles urea. So that's number 1. Okay. So I think it helps to draw a picture. So I'm going to draw a beaker, right? And I'm going to draw a red blood cell. So here's my red blood cell. Okay. Now, what do I know about this beaker? Well, I know a couple things. I know that this is 360 milliosmoles on the outside, right? And then I know that the red blood cell, to start out, as all human red blood cells are, it's 280 milliosmoles inside, okay? So number 2 tells me determine the osmicity. And to determine the osmicity, I compare the solution at 360 to the cell at 280. So 360 is more than 280. All right, so 360 more than 280. So this is going to be hyperosmotic. All right, so we're doing the osmicity, so you're always going to have the word osmotic, right? That's the initial condition of the cell, so it's hyperosmotic. Okay, so that's number 2. Number 3 says, move urea if you have it. Now we know this is urea, and what's going to happen is urea, because there's no urea inside a red cell to start, it's going to diffuse into the red cell. And as urea diffuses into the red cell, because it's permeable in the cell membrane, right, it's going to make the inside of the red blood cell have a higher osmolarity because it's moving particles in. And consequently, as I move every particle from outside to inside, I have 1 less particle outside. So the easiest way to do this is just divide urea in half. All right, so half of the urea is going to go inside. So half of 360 is 180. So I still have 180 left on the outside. But now I have 280 plus 180. All right, does that make sense? So that gives me, for the inside of the red blood cell total, the 280 I started plus the 180 I have now is 460 milliosmoles. So 460 inside, right? The 280 stuck inside, half of the urea diffused in, so now the inside is 280 plus 180. Make sure I did my math right, is 460, and outside is 180, okay? Now I want to show the direction of water movement. So you draw an arrow to the biggest number, 460 inside the cell, 180 outside the cell. So my arrow is going into the cell, right? So my arrow is going into the cell, which means the direction of water movement is going to be into the cell. And because water is going into the cell is going to get larger. Cells get larger in a hypotonic solution. So even though this was hyperosmotic, its tonicity is hypotonic. And that's how you do these problems. All right, so that's one way to do it. And I think drawing a picture really helps. It's hard to screw up. It helps you keep track of things. And so hopefully, that little example helped a little bit there. We're going to do one more, and we'll do it like we did the first one, though, without the drawing. So I think we can do number 6. All right, we'll look at number 6. Remember that the osmolarity is due solely to the number of dissolved particles. So we can add particles together, and as long as we know what the molarity or osmolarity is of each one, we can go ahead and figure those things out. So in this case, in number 6, we have 140 milliosmoles of urea. We're already in milliosmoles, so we don't have to do anything to it. And 140 milliosmoles for glucose. So all we have to do is add them together, right? So 140 and 140 is 280. So our solution is 280 to start. Our red cell is 280, as human red blood cells always will be. So I compare the solution to the cell. It's like we had the first time. Same, it's isoosmotic. Now in this case, urea and glucose, right? They're separate particles, right? Urea is urea and glucose is glucose. But one of them can penetrate the membrane in terms of urea, right? Glucose is stuck. Remember, glucose can't penetrate, nor can either any of the salts, sodium, potassium, or calcium chloride. They're all stuck, but urea can penetrate the membrane. So just like we had before, half of the urea is going to diffuse in, half's going to stay out. So half of 140 is 70. So 70 is going to go inside the red blood cell and make it 350. We have to subtract 70 from this 280 because it went inside, so that leaves us 210 on the outside. So once we move urea, we're going to have 210 outside and 350 inside, because that's 280 plus 70. All right, I guess it'll be right here. 350 here. So what you do then is you draw an arrow to the biggest number, which will be into the cell. Because the cell is going to pick up water and get larger, it's going to be a hypotonic solution. So you should practice all these on your own and kind of look to see how that one works. That's number 4. Number 5 looks at Fick's law of diffusion. And Fick's law of diffusion explains how the diffusion rate moves across a membrane. So in order to apply Fick's law of diffusion to a situation, it must fit the criteria that the particle is small and nonpolar and can move across the cell membrane. Okay, so that's number 1. We want to know both qualitatively and quantitatively what's happening. So qualitatively is saying, okay, is it going to affect the net flux? Remember, net flux is the appearance of the particle on the other side of the membrane. Okay? And it can do one of 3 things. It can either have no change in the net flux, it could have an increase in the net flux, it could have a decrease in the net flux. That's the qualitative change. Quantitatively, we can estimate the actual change by understanding, right, the numbers. And we're going to make it simple in this class, and we're going to only change one variable at a time. All right. So here's Fick's law of diffusion, right? You should make sure you understand this for the exam, right? And basically, there's 2 equations we have to know for the lecture exam. For the lecture exam, I never give you an equation sheet. I expect you to be able to figure those out on your own. And we'll get more complicated as the semester goes. But for the first exam, the 2 formulas you have to know, we already did, right? One is the millimolarity times the number of associable particles. We just did that the last problem. And the other one is this one, Fick's law. And Fick's law tells us the net flux rate, the appearance of the molecule on the other side of the membrane, equals the surface area, so the available surface area, times the concentration gradient. Now, these are directly proportional, right? They're above, so I'll just highlight those. They're above the divided sign, right? Which means that if the surface area goes up, flux goes up. If the concentration gradient goes up, flux goes up, which makes sense, right? If I have a bigger area in which I can strike the membrane and move across, that's going to increase how fast things go. And if I have more particles, it's more likely it's going to hit the membrane and go across. So that's the first part, surface area times concentration gradient divided by membrane resistance times membrane thickness. So notice these two factors, the membrane factors, are on the bottom, which means these are inversely proportional to the net flux. If membrane resistance goes up, flux goes down. Membrane thickness goes up, flux goes down. Okay? So when we do these problems, let's take a look at them and kind of see how it works. So here's your first example. An experimental drug is given that increases the membrane resistance to oxygen of mitochondria by 4-fold. What happens to the diffusion rate of oxygen into the mitochondria? So the first thing you ask yourself is, does oxygen fit that criteria of being able to move directly across the membrane, right? And again, small, yeah. Nonpolar, yeah. Oxygen's great. Oxygen moves across the membrane by diffusion all the time. That's how we live, right? So it's going to go across the membrane by itself, so it fits Fick's law. So what we're going to do is we're going to write out Fick's law, right? Available surface area times concentration gradient divided by membrane resistance times membrane thickness. And then look at the equation and say, okay, what happens? The membrane resistance, right, clearly identified here, goes up 4 times. So here's our equation, and right here we're going to highlight membrane resistance. So we're going to know that goes up 4 times. It said nothing about the other 3 variables, so we assume they haven't changed. So quantitatively, right, we'll use those numbers, right? So we'll look at this and go, okay, available surface area, that hasn't changed, we're going to make it a 1. Concentration gradient, that hasn't changed, we'll make it a 1. Membrane resistance, right? Sorry, membrane resistance, right? That's the one that went up. You can see it right here, membrane resistance in the problem. That went up 4-fold, right? So here's our membrane resistance in the problem, highlighted. And here's my 4-fold, highlight that. So that becomes 4. Doesn't say anything about thickness, that stays 1. And if you do that math, you get 1 times 1 is 1, divided by 4 times 1 is 4. That equals 1/4. So the net flux will go to 1/4. Now we should also look at it qualitatively. That was quantitative. Qualitatively is this, should it go up or down? Well, since membrane resistance went up and it's inversely proportional, then flux should go down. And take it a step further, just kind of check yourself. If flux goes down, this number should be less than 1. Right? So if you expect flux to go down, the net flux rate should be less than 1. If we expect flux to go up, the net flux rate should be greater than 1. All right. So we'll just look at 2 more and then let you figure out the other 2 on your own. So here's B. An extremely rare genetic disease triples the thickness of the cell membrane of a nerve cell. What happens to the diffusion rate of sodium through the membrane? Okay. So you should look at this and go, well, sodium, well, wait a minute. You're trying to trick me. Because sodium, while small, is polar. Sodium's going to be Na+, right? It's an ion. And so since it's a polar molecule, it doesn't go through the membrane. So in reality, there is no change in the flux because it can't go across the membrane by itself. So B is so easy, it's hard, if that makes sense. So let's look at C. So C gives me hyperbaric oxygen therapy, which increases the partial pressure of oxygen, is given to a patient that doubles the concentration and partial pressure of oxygen in the blood. What happens to the diffusion rate of oxygen into the pulmonary capillaries? So that means oxygen is going from the lungs into the pulmonary capillary. So it's going to go directly across the membrane. So again, we write out our equation here, our same basic equation we had, and then we just replace the number. In this case, the number it gave us was doubles the concentration. Okay? So everything stays 1 except for concentration gradient. So I get 1 times 2, that's my double the concentration, divided by 1 times 1. So 1 times 2 is 2, divided by 1 times 1 is 1, 2 over 1 is 2. So my net flux rate goes to 2. That's my quantitative part. Qualitatively, you should look at it and say, okay, well, it's greater than 1, so it should go up. And then I always look and do that double check. Well, since it's concentration gradient, it's on the top here, right? These two on the top, means it's directly proportional to flux. So if concentration goes up, flux should go up. So qualitatively, flux should go up quantitatively by 2. All right. So I'll leave you for D and E to work on your own, and F as well, and G. All right, so get a chance to work on some of those your own. Here's the good news for you, right? We're not going to do number 6. Why aren't we going to do number 6? Because remember we said that the enzyme activity, so that's S1P5, section 1, part 5, is not on the lecture exam. So we're not going to worry about this. This might help if you're studying for the checkup that has this information on it, but this is not on the lecture exam. So let's skip to the bonus material. If we were in a face-to-face class, we probably wouldn't do these. We probably would have in this case, because one group that would normally got 6, we'd give them 7. I think there's an 8 too, right? So let's kind of look at these ones. These are a little shorter, but they're important. So basically what number 7 asks is to compare and contrast the 3 major types of muscle: skeletal, cardiac, and smooth. And what I like to do is I like to put cardiac in the middle. I like to put it in the middle for a couple reasons, okay? In many ways, cardiac looks a lot like skeletal, but behaves a lot like smooth. And obviously it has its own attributes as well. So when we look at the different types of muscle, right, the first question is, do they have striations? And the answer for skeletal and cardiac, again, they look similar, so not surprisingly, they both have striations. Smooth muscle doesn't have striations. As a matter of fact, smooth muscle is named smooth muscle because it lacks striations, so it's smooth under the microscope. The control. Now, all muscles have the ability to be reflexive, but skeletal muscle is considered to be voluntary. You want to move your hand, you can, as an example. While cardiac muscle and smooth muscle are involuntary. You don't have that level of control. Shape, what does it look like? Skeletal muscle is long and cylindrical. Cardiac muscle is short and chubby. And smooth muscle, in books I always say spindle shape. What does a spindle look like? Cigar. So think of a cigar shape, right? So long, skinny, tapered at the ends a little bit. Nucleate, I mentioned in an earlier video. Skeletal muscle has multiple nuclei, so it's multinucleate, while cardiac and smooth are uninucleate. They have 1 nuclei each. In terms of contraction, we need calcium to bind to muscle, no matter what type of muscle it is. So where does the calcium come from? Well, skeletal muscle, it's all stored inside, intracellular, in the, particularly for skeletal muscle, in the lateral sacs of the terminal cisternae, the sarcoplasmic reticulum. Calcium comes from intracellular and extracellular means for cardiac muscle, so it's sort of a combination of both. And smooth muscle is extracellular only. So we open calcium channels, calcium moves in and causes contraction in smooth muscle. Right? There's some component of that in cardiac muscle as well, where we have a little bit of storage of calcium and also calcium channels, which determines the strength of contraction in cardiac. Skeletal is all stored calcium inside the cell that leads to contraction. What's the calcium binding structure? So in order to get contraction, calcium has to bind to some protein. It's troponin, or some people say troponin, in skeletal and cardiac muscle. And in smooth muscle, it's a molecule called calmodulin. It modulates activity by calcium. That's where the word comes from. Right? Skeletal muscle has a very short refractory period, which means it can recover and fire again very quickly. Cardiac muscle is a very long refractory period. It means it has to have a long period before it resets and can fire again, which is actually a very good thing from a functional standpoint. And then smooth muscle has a very short, not a very short, a short refractory period. Not quite as short as skeletal muscle, but not nearly as long as cardiac muscle. Cardiac muscle, as an example, in the ventricular area, has a refractory period that lasts up to a quarter of a second. So you can only contract the heart so fast, and there's always an opportunity where the heart can't contract again. That's good because the heart needs to fill with blood to pump it. And so, as an example, your heart can't go into tetanus. It can't go into this prolonged contraction. And that's good because if it did, you would die, right? So, if your heart's not pumping blood, you're gonna die very shortly. So that's refractory period. Contraction control, because it's voluntary. Skeletal somatic nervous system. And the cardiac and smooth muscle have very different contraction control parameters, and it's different for different things. As an example, cardiac muscle and a few types of smooth muscle are autorhythmic. They can contract on their own, as an example. Contraction duration, skeletal muscle is very short, cardiac's fairly short, and smooth muscle is relatively long comparatively. So those are kind of the different characteristics of the three different types of muscle. I also added the muscle twitches here. In order for a muscle to move, the tension developed must exceed the load. All right, and that's especially true of shortening. We call those contractions, and muscle contractions can occur in a variety of ways. You can divide it up into three basic types. The first type is called isometric. Iso, same, metric, length. So isometric means you develop tension, right? But there's no change in length. So an isometric contraction would be like take your hands, clasp them together, and pull as hard as you can apart, right? Nothing moves, right? That's called an isometric contraction. You feel your chest and your arms tighten up, but nothing moves. Isometric. If it's not isometric, it means it's isotonic. Isotonic contractions, that means constant tension, right, is when the muscle changes length during contraction. And there's two types of ways we can change length. One is we can shorten, and the other is we can lengthen. So when we shorten a muscle and cause contraction, that's called a concentric, towards the center, towards the middle. So a concentric contraction is when a muscle shortens during contraction. And if the muscle lengthens during contraction, it's called an eccentric. Eccentric means away from the middle. Okay? So eccentric contractions lengthen muscle, concentric contractions shorten muscle, and isometric contractions have no change in muscle length. So that was the bonus number 7. Here's bonus number 8. So explain the channels, and just, again, to make sure you understand what channels are. There are channels within the cell membrane that are integral proteins that allow especially relatively small molecules that are charged to be able to move from one side of the membrane to the other. When we divide channels up, we can divide them up into a couple different ways. The first major way is what we call non-gated channels versus gated channels. A non-gated channel is an opening that extends across the membrane where there's no gate on it, meaning that it's always open. And so that means that as long as we have a concentration gradient from one side of the membrane to the other, that molecule is going to constantly diffuse through because there's no control over it. Okay? There's relatively few of those channels in the human body, but they do exist, and they are important for maintaining certain things in terms of electricity, and especially in nerve and muscle cells. Since relatively few are non-gated, that means most are gated channels. And gated channels gives us much more selectivity over when we move materials from one side to the other. And the only time materials move is when there's a passageway, there's no gate there in the way, no doorway, if you will, to prevent the movement from one side of the membrane to the other. Now, the vast majority of gated channels have a single gate, one gate only. There's a special type of double gated channel we see for sodium in muscle and nerve. And those are actually what causes the action potential. So we'll get more of that later on in the semester. So there's one type of double gated channel, a few types of non-gated channels, and everything else. All the other channels are gated channels that have one and only one gate. Okay? Now, in order to open or close that gate, or what they call a change in the channel configuration, meaning if the gate was open, it is now closed, or if the gate was closed, it is now open. There are 6 different ways we can get that channel to change its configuration. Okay? Now, generally speaking, each specific gate has a specific way it opens or closes. So there's not a gate that does all of 6 of these things at once, but there are some gates that open one way or close another, or might have kind of an extra additional way to open or close that kind of serves as a safety factor. Okay, but most of these are what we call not multimodal, meaning they have one mode that excites them or causes them to close a gate. Okay, so one type is a chemically gated channel, and that means a ligand, some sort of specific chemical, binds to a receptor, and that opens or closes the channel. We have voltage gated channels, voltage gated, right, which means it's determined whether the channel opens or closes by the electrical state of the cell. So a certain voltage goes through the channel and it opens, or a certain voltage goes through the channel and it closes. We have mechanically gated channels. Those change their configuration based on some sort of physical force. So like a pressure channel or a touch channel would be examples of those, where there's some sort of physical thing that disrupts the channel. Time-gated channels open or close based on some specific unit of time. So some time lapse has to occur before they change their configuration. We have time-gated channels in our neurons that close up after a certain amount of time, which then leads us to a refractory period. So we also see it, like we just talked about that, with the different types of muscle. So time-gated channels close at specific amounts of time. We have light-gated channels, channels that open or close at exposure to photons of light. Photons of light are the units of light. We have them both in our forehead and in our popliteal folds. That's the back of the knees. And those help to maintain what we call diurnal variation, that keep our body in that day-night cycle. So it's light-gated channels. And the last are temperature-gated channels. And temperature changes will cause channels to open or close. So those are sort of the big types of channels. All right. So that's the end of the information there. And again, just to kind of explain a few things. The information that we presented probably covers in the 2 videos approximately half the material on the exam. So there are things certainly you should know, but you need to study your notes. You need to study the text-based notes. You need to look at the slides. Hopefully you've already listened to the videos and taken notes, so you don't have to go back and do those, and you already have the information you need, and you need to study these group problems, right? And I put the 3 biggest things as kind of a tie for first is the text-based notes, and then your lecture notes you took from the videos. A very close second, I guess if it's tied for first, it would be a very close third, would be the group problems that we're just finishing up right now. And the 4th thing would be the slides. The slides are good if you're a visual learner. The problem, though, again, is that they don't have the same detail and enrichment that the notes and the videos have. And so if you just knew the slides, you would have a basic idea of things going on, but you probably wouldn't be able to answer some of those detailed questions, right? You're also going to be asked to apply some knowledge. So make sure you understand how to apply the information to a new situation. Not every question will be like that, but there certainly will be. So that's gonna end this video, and good luck.